Hi guys, welcome to Med Nuggets channel. Today we are going to see about laboratory diagnosis of parasitic infections of GIT. There are many infections caused by parasites in GIT, but now we are going to see about some of the important parasitic infections caused by parasites in the GIT. To start off with the parasitic infections, laboratory diagnosis, first we need the specimen, right? Maximum the specimen collected will be of stool specimen. In some other cases, it may vary. And in all laboratory diagnosis, you can include antigen detection, antibody detection, and molecular methods. And don't forget that. Now, let's start with ascariasis. Ascariasis is an infection of the small intestine caused by Ascaris lumbricoides. Now, laboratory diagnosis, we are going to do stool examination. In stool, you can detect both fertilized and unfertilized egg in saline and iodine wet mount. There are some differentiating points between fertilized and unfertilized eggs seen in the wet mount. Okay. Now let's see about the unfertilized egg. The albuminous coat covering the unfertilized egg will be of thin albuminous coat, and the shape will be elongated in shape. And it contains an unsegmented ovum, and it will be of atrophied ovum, and there will be no presenting space at the poles. The eggs will be bile stained so that it appears golden brown in the saline mount, and it does not float in the saturated soil solution. More or less, unfertilized egg and the fertilized egg will be of same features, but something it may vary. Now let's see about the fertilized egg. Fertilized egg, the albuminous coat of fertilized egg will be of thick albuminous coat, whereas in an unfertilized egg, the albuminous coat is thin, and the shape of the fertilized egg will be round to oval. And both in fertilized egg and unfertilized egg, the ovum is unsegmented. But in unfertilized egg, there is no space at the poles, but whereas in fertilized egg, there will be a clear crescentic space at the poles. At both the poles, there will be a crescentic space. The eggs will be bile stained, appears golden brown in saline mount, and it floats in saturated salt solution. Here you can see the picture of fertilized and unfertilized egg, and as of I have told you before itself, both are bile stained and appears golden brown in saline mount. And you can see adult form by naked eye or barium meal X-ray of GIT, and antigen detection, molecular test, eosinophilic condition will be present, and you can see presence of charcoal leaden crystals in sputum and stool. Now let's see about intestinal teniasis. First we can see about intestinal amoebiasis. Now we can see about intestinal amoebiasis. Amoeba is the single-celled protozoan parasite that constantly changes their shape, which is due to the presence of an organ of locomotion called pseudopodium, which means there will be an organ called for locomotion, pseudopodium will be present. Because of that, it will constantly change their shape. Now let's see the laboratory diagnosis. The specimen collected will be, you should collect minimum 3 stool samples on alternate days. And you can collect rectal ulcer tissues or rectal exudates. Now let's do the stool macroscopy. The amoebic stool will be of foul smelling and copious in amount. And then color will be dark red in color mixed with blood and mucus. Now I have told you that stool macroscopy, it will be a foul smelling and it will be dark red colored mixed with mucus and blood. You can use concentration techniques by using formalin ether sedimentation method. And then permanent staining is done by using trichome or hematoxylin stain, so that you can examine the stool. Now we are going to demonstrate the trophozoites. You can see trophozoites in the freshly passed stool. Demonstration of trophozoites is considered as the gold standard microscopic test for active infection. They are motile with finger-like pseudopodias. And then hematophagous trophozoites, you can see RBC in their cytoplasm. And then nucleus of the trophozoites will be of cartwheel appearance. And then there are many cyst stages available present, and then in the uninucleated cyst it contains chromatoid bodies and a large glycogen mass which stains brown with iodine. Here is the picture of trophozoites and the quadrinucleated cyst. Cyst is the infective and the diagnostic form. And then now we are going to do the culture. For culture you can use polyzenic culture media, which is known as NIH media, or else you can use axenic culture media, which is also known as Diamond's medium. And serological test is also done. You can detect the amoebic antigen, amoebic antibody, isoenzymes, and nested multiplex PCR, real-time PCR, biofire film array is also done. Serological test is considered as the gold standard method for the diagnosis. Now let's move on to the giardiasis. Giardiasis is the infection caused by Giardia lamblia. It can be Giardia intestinalis or duodenale. Now let's move on to the laboratory diagnosis. We are going to do stool microscopy, which is the gold standard method for the detection of the infection. It is used for detection of cyst and trophozoites. Tetranucleated oval cyst is the diagnostic and infective form, so we are going to detect that in the stool microscopy. Trophozoites, you should take at least 3 samples on alternate days for examination of trophozoites. They adhere firmly to intestinal mucosa by adhesive disc causing intermittent shedding. Trophozoites from the front view, it will be appearing as pear-shaped or teardrop or tennis racket appearance, and from laterally it appears as spoon or sickle-shaped. Motility of the trophozoites will be a falling leaf-like motility. Here is the picture. You can see the tennis racket or the teardrop shaped appearance of the trophozoite from the front view. In diagram B, you can see the lateral view. And then no pus cells or RBCs will be seen in the cytoplasm of the trophozoites, whereas hematophyte trophozoites in the amoebiasis, you can see the RBCs in the cytoplasm. Here also you can use the concentration techniques like zinc sulfate flotation or formal ether sedimentation method you can use for concentration of the stool. By the help of duodenal sampling, you can do entro or string test. And then as usual, antigen detection, antibody detection, molecular methods are done. And radiology, you can use X-ray taken after the barium meal. It's non-specific, irregular mucosal thickening with dilated loops of hypotonic bowel can be seen. Culture used is Diamond's media you can use, which is the axonic culture media. And I have told you before that you can see in radiology also X-ray taken after the barium meal. Giardia will be inhabited in the crypts of duodenum and upper part of jejunum, which can be seen as mucosal thickening with dilated loops. And remember that trophozoites can be seen only in the active infection. Active disease, you can see this. It shows antigenic variation also because of cystine presence. Cystine is rich over there. And now let's move on to other parasitic infection, intestinal teniasis. Teniasis is the infection caused by Tinea saginata or Tinea solium. Now let's move on to the laboratory diagnosis. The collected specimen is stool. In stool examination, you can demonstrate the egg, and less often proglottids can also be seen. We are going to do wet mount. In wet mount, you can see eggs or proglottids. Concentration techniques can also be used to increase the detection rate. Eggs. Actually, the eggs of Tinea saginata and solium will be morphologically similar. They are round, bile stain. Bile stain means they appear golden brown in the mount. And they do not float in the saturated salt solution. It contains the embryo with 6 hooklets. Embryo is covered by embryophore. In that embryo, you will be having 3 pair of hooklets, so that 6 hooklets will be present. There will be a eggshell covering the embryo. Embryo will be covered by the embryophore. Inside that you will be having 3 pair of hooklets. Totally 6 hooklets will be present. The larva is also present in the teniasis infection. Cysticercus bovis in Tinea saginata and Cysticercus cellulacea in Tinea solium. The adult will be having suckers for attachment. And proglottids can also be seen. Antigen detection, coproantigen can be detected, and molecular methods is done targeting the mitochondrial DNA, which can distinguish the Tinea species. And I think so intestinal teniasis is also clear for you guys. Now let's move on to the Hymenelepsis nana. Hymenelepsiasis. Hymenelepsis nana is also known as dwarf tapeworm. Man acquires infection by the ingestion of food and water or else by the auto-infection. Now let's see the laboratory diagnosis. The Hymenelepsis nana, stool microscopy, you can detect the egg. Egg will be of round to oval in shape. The egg will be non-bile stained, so that colorless in the saline mount. 2 membranes will be surrounding an embryo. The same like teniasis, here also Hymenelepsis nana contains 6 hooklets. That means 3 pair of hooklets. And you can see the presence of polar filament at both the poles of embryophore. And then the egg is the infective and the diagnostic form of the infection. And the larval stage is known as cysticercoid larva. See, embryo is covered by the inner membrane and the outer membrane. and you can see the presence of polar filaments at both the poles of the embryophore, which I have drawn blue in color, and you can see the presence of yolk granules, which I am drawing in the purple color, and you can see the presence of 6 hooklets, 3 pair of hooklets. For all parasitic infections, maximum the prevention will be maintaining the good personal hygiene. And in some cases, salting and pickling can be effective, whereas in teniasis, salting and pickling won't be effective. You have to refrigerate at 4 degrees Celsius. Whereas here, good personal hygiene and improved sanitation can control the disease. And then another species called Hymenelepsis diminuta, which is also known as rat tapeworm. There will be no polar filament present and the eggs will be of bile stained. Hymenelepsis nana is known as dwarf tapeworm, whereas Hymenelepsis diminuta is known as rat tapeworm. And then totally opposite, Hymenelepsis nana, there will be presence of polar filament and non-bile stained egg, whereas here no polar filament and the eggs will be bile stained. I think so Hymenelepsis nana infection laboratory diagnosis is clear to you guys. Now let's move on to another parasitic infection, schistosomiasis. Schistosomiasis is caused by Schistosomiasis japonicum, Schistosomiasis mansoni in GIT, whereas in the venous plexus of bladder, Schistosomiasis hematobium causes the infection. Now let's see the laboratory diagnosis. Stool examination, you can see the egg. The eggs of Schistosomiasis mansoni will be oval, elongated and non-operculated, and there will be presence of rudimentary lateral spine. Sorry guys, Schistosomiasis mansoni will be there presence of lateral spine. Rudimentary lateral spine will be present in Schistosomiasis japonicum. You guys also don't confuse like me. Schistosomiasis mansoni, there will be presence of lateral spine, whereas in japonicum it will be rudimentary lateral spine. See, you can see the presence of spine, Schistosomiasis egg in the picture. In hematobium, you will be having the spine in the middle, whereas in the mansoni lateral spine, japonicum, it's rudimentary. The eggshells of mansoni will be acid fast. And then hatching test is also done, which causes hatching of motile Miracidia larva from the egg. The larval stage is known as Miracidia larva, which can be seen when stool specimen is diluted in water. Thereby confirming the viability of the parasite by hatching test. And then antigen detection is done by detecting the circulating cathodic and anodic antigen in the serum and urine. And antibody detection is also done. It's less useful because the cross reaction can occur. And then drug of choice for treatment is praziquantel. Prevention includes maintaining the personal hygiene and proper disposal of human excreta. I think so Schistosomiasis is also clear to you guys. Blood fluke, Schistosomiasis. Now let's move on to the another parasitic infection, trichuriasis. Trichuriasis is caused by Trichuris trichuria, which is also known as whipworm. The diagnostic form here is unembryonated egg in the fecus. Here it will become embryonated only in the environment, which is the embryonated egg is the infective form. Here we are going to do the stool microscopy. The eggs of trichuriasis will be barrel shaped with mucus plugs at the end. It will be of bile stain so that it appears golden brown in the saline mount. And then the eggs will be floating in the saturated salt solution. And you can also see adult worm on proctoscopy. The adult worm will be of whip shaped. And then the adult worm anterior 3/5 will be of thin hair like and it will be of coiled appearance. Anterior only coiled. Posterior it will be of short and thicker 2/5. Anterior is longer but it will be coiled, but posterior is somewhat shorter than anterior but it will be thicker in appearance and short. Now let's move on to another parasitic infections, enterobiasis. Enterobiasis is an infection caused by Enterobius vermicularis, which is also known as pinworm or threadworm. Transmission is by contaminated fingers or by autoinfection. Now let's see the laboratory diagnosis. Usually the specimen will be perianal skin samples. The specimen is collected by two methods. One is cellophane tape or NIH swab method. Cellophane tape is applied onto the perianal skin and then tap is mounted with a drop of saline. And the number of specimens collected should be 4 to 6 as female worms migrate intermittently. And then the sample should be collected late evening or early morning. The timing should be maintained. Should be late evening or early morning because it has the more chance of egg deposition. The eggs of Enterobius will be of palano-convex in shape. One side it will be of convex and other side it will be of flat. While doing NIH swab or any other sample collections, these things have to be kept in mind. One is number of specimen collected should be of 4 to 6 and then timing should be maintained, should be of late evening or early morning. And then the eggs of Enterobius, you can see it will be palano-convex in shape. One side it will be of convex and other side it will be flat. Embryonate egg will contain the See guys, cellophane tape method is also done. Embryonate egg will be of non-bile stain, colorless. Floats in saturated salt solution will be of tadpole shape. Larva will be present. Now let's see the hookworm infection, which is one of the important cause of iron deficiency anemia. The species causing this infection are Encyclostoma duodenale and Necator americanus. Both the species will be having egg and first larval stage somewhat same appearance. And then while doing stool microscopy, the eggs will be of oval in shape, surrounded by thin eggshell. The eggs will be of non-bile stain so that appear colorless in the saline mount. And ovum is segmented containing 4 blastomeres. It floats in saturated salt solution. You can't differentiate between the eggs of Encyclostoma duodenale and Necator americanus because it will be indistinguishable, morphologically indistinguishable in the saline mount. There will be a clear space present between the eggshell and the embryo. Remember guys, there will be 4 blastomeres present in the ovum. And then egg counting is also done. Number of eggs per gram of stool, Cat-O-Cat's technique. Stool culture can also be done in the agar plate technique. And Harada Mori filter pepper tube method is also done. Sorry guys, just remembered I haven't covered strong alloys yet. I will add it as a shorts. Thank you guys for your support. Please do like, comment and subscribe. Bye bye guys.